Welcome to Eureka. I'm Gauhar Raza and you are watching Eureka. This is a program where we bring you face to face with an outstanding scientist. Let me begin by a story once again. There was a young scientist in Australia. His name was Barry Marshall. The young scientist didn't have animals to test his hypothesis, and his hypothesis was very simple. He thought that acidity and ulcer is caused by a pathogen, a small bacteria. He couldn't test his hypothesis anyway in the laboratory. He asked one of his colleagues to extract this small bacteria, mix it with extract of beef, and he drank it. 3 days later, he fell sick. The mother told him that he was smelly. And then, after 20 years of this incident, Barry got Nobel Prize. Following in the same footsteps, there was a young scientist who somehow was interested in chemistry, but there was a turn in his life and he started working on the same pathogen and got Indian Nobel Prize known as Bhatnagar Award. Welcome, Dr. Suman.
Thank you.
We are privileged to have you here. But let me begin by asking you the same question, which everybody has in mind. When did you start working on this pathogen?
Helicobacter pylori, H. pylori.
H. pylori, as it's generally known as.
As a matter of fact, I was infected with H. pylori when I was a student.
So was I.
So during my PhD, it was really bad, like 2 years almost. I was diagnosed, and this bacteria, you can get reinfection also. So once I was treated, and then after that, after another year, I got sick again with this infection. So, but I don't think that was the reason that I started working on this bacteria, because that was around 93, 94. So I finished my PhD and then went to Harvard Medical School for my postdoc.
Right.
And there I worked on mammalian DNA replication. And after 3 years, I got a job in JNU. And I was supposed to start my own lab. So I was thinking of starting something which will be relevant for India, as well as I can use my expertise, what I have learned.
Acidity is more of a problem of developing countries. It's much lesser in developed countries, but they're catching up. That is what the data says generally.
Yeah, but H. pylori, almost 50% of the world population
Lives in.
is actually infected with H. pylori.
Right.
And it's all over the world.
Correct.
And the reason, as I said, when I was planning to come back, I was thinking what I should start on. And during that time, 2 strains of H. pylori were sequenced. And that was the first bacteria, because of its importance.
Right.
2 strains were sequenced. These are 26695.
But it's probably the oldest friend of human beings also.
Right, right.
About 50,000 years ago, human beings got infected.
Yes, and not only that.
And it has also played a very important role in human development, because if human beings didn't have this bacteria in their stomach, they would have probably faced a massive problem when they migrated from Africa.
Could be, but at the same time, the niche of this bacteria, if you think, is unparalleled. It's in the gastric lumen, where the pH is around 3.
It's very high.
No other
bacteria can survive
there. So this bacteria is so clever.
That's why probably a number of scientists and doctors always thought that acidity is because of hypertension.
Yes.
That it couldn't be a bacterial infection.
Right. And as you said rightly in the beginning, why he had to inoculate himself? Because nobody believed him. And if you recall Koch's third postulate, that if you isolate a pathogen, you have to prove that the pathogen is causing the same disease.
Right.
And there is no other way he could convince the world that, see, this is what has been taken away from a patient, cultured, and now if you re-inoculate, you get the disease.
Absolutely.
But there are many other factors, like even though 50% of the world population harbored this, disease may be 2 to 3% maximum. The reason being the host immune system, host genetic makeup. As you know, like even if somebody sneezes, not all the people in that room are going to get infected.
Right.
It also depends on how much resistant you are. And this bacteria is very clever because it has an urease enzyme which breaks down urea and forms ammonia. And this ammonia neutralizes the acid environment.
Right.
And then it goes past this thick mucus layer.
Right.
And it reaches the epithelial border.
Which basically protects our system, stomach especially,
from this
from the
acids,
pH,
yes, from this pH.
high pH value.
Right.
Yeah. I would like to ask you a very natural question. A lot of people must have asked you this question in life, that what exactly you did to deserve such a high award? Because Marshall had already done work.
Yes.
For 20 years.
Right. That's true. Very recently, last week, there's a recent paper, people were thinking how H. pylori causes cancer. And people have shown now, scientists have shown, that H. pylori causes double-strand DNA break. And as you know, if you have a DNA break in your system, that definitely is
triggers
many of these phenomena. So we were trying to understand that how this bacteria multiplies, because multiplication is also one process of pathogenicity. Once the pathogen enters the body, it has to multiply. Unless it reaches a number, stable. So we looked at the genome when the genome sequence came up, and we found there are many proteins which are very obvious in known system like E. coli or Bacillus. Apparently they are absent in this bacteria. One of the major protein is a helicase. And what is the helicase? Helicase is the enzyme which is required for the separation of the two strands of DNA.
Absolutely, yep.
There is a protein which called DNA C, which is a helicase loader, because helicases cannot be loaded on the DNA by itself. You need a loader.
Right.
When you looked at the genome of HP, we found that there is no DNA C-like protein.
And it was an important observation.
It was an important
observation. Which called for a major recognition of your team. We'll continue the discussion. I have to take a break. Don't go anywhere. We'll come back soon. Welcome back. Dr. Suman Dhar. You come obviously from, your name it's apparent, you come from West Bengal.
Yes.
Belong to a middle class family. Your father was a government servant. When did you start your interest in science and decided that this is what I'm going to pursue, and why did you do that? Was it because of the pressure from the family? Was it your interest? What was it, because of a teacher or a school was pushing you towards science? What happened?
No, I don't think.
Or you had a special interest in chemistry?
I did. Actually, I must tell you this, I'm colorblind, and that too red and green colorblind, which I did not know.
One Indian among 5 is colorblind.
And I'll tell you, I liked chemistry very much, and nobody pressurized neither. And during those days, although it looks like I'm very old, but at least even then also, there was not much pressure. Like you read and then you do whatever you want to do, but do it well. So after high school, two things happened. So I took admission in chemistry, but then I realized that I'm severe colorblind, and that to red-green. And as you know, in chemistry practical, you have to do this flame test.
Right.
So my teacher would ask me that differentiate between these elements based on different shades of red, which is crimson red, scarlet red, but the person who doesn't even know what is red, how do you know?
You wouldn't have been able to distinguish
No,
between
nothing,
all
absolutely
these colors.
nothing,
and you couldn't do the practicals.
I couldn't do, and all my friends, they will stand and I will do the titration. Invariably, I will fail, but the important thing was you have physical chemistry where you don't have colors. You can
Right.
do surface tension, you can do viscosity.
Right.
So actually, I passed because of physical chemistry. There was no color. But then, as I really liked and I wanted to continue, and I lost my father also.
When you were 10 years,
Yeah,
when
when-
you were in 10th class.
plus two. So I realized that I have to work hard and I have to do well. And then I found biochemistry is
It would
very
have been a
fascinating.
very tragic and traumatic experience that your elder brother had to leave
Yes, because
his studies
yes,
and join a
right.
job,
Because as
take
you
up a
know,
job.
as a lower middle class family, what happens is
Yep.
if the earning member somehow is not there, then somebody has to come and then take care of the family. So he actually did a lot for the family, and he is still continuing. And I'm really, really thankful to him and my family members. But then I found that in biochemistry, you don't have to look at the colors by yourself.
You had technology which came to your
Right,
rescue.
you have spectrophotometers, which will read the colors for you.
Yes.
So that's how I started studying after my chemistry honors, biochemistry. And I did that in Kalyani University.
But for a spectrometer, we would have lost an outstanding scientist.
I think it's the question what you ask and pursue it. I think even now I am thinking that not only the asking question, if you believe in something, you have to pursue it. So if I would have continued in chemistry, maybe I would have asked something else. And since I am here, I am asking those questions which to me are relevant, and I can add something.
That brings me to something else, which is I have been very curious about. Is you were working on Plasmodium.
Yes.
And you were working on something that resides in the stomach. One of these affects liver, the other one stomach. What is the relationship between the two? This is apparently there's no relationship.
But both affects human. Both are pathogens.
Both are pathogens.
And so
one causes malaria,
one
the other
causes
one
malaria,
causes...
one causes gastric ulcer, gastric adenocarcinoma. So the malaria, the way we started is more because I also have interest in looking at DNA replication in higher eukaryotes. As I told you that my postdoctoral studies were mostly on mammalian system and how this uncontrolled cell proliferation takes place in cancer. Those were the questions we were asking. But after coming back, I was thinking that I should also study some pathogens which are more related to eukaryotes, which is a nucleus, other organelles. And Plasmodium fascinated me. The reason being, you mentioned about liver. Of course, it goes to the liver.
Correct.
But the fever which you get is for the erythrocyte blood stage.
Right.
So it goes to the RBC, it infects, and then it multiplies tremendously.
And suddenly comes to bloodstream.
And then from one infected RBC, you can get 16 to 32 new parasites.
So something triggers this multiplication, which is large in number, very, very large in number.
And this continues. And this fever which you get is basically due to the sudden burst of RBCs and taking out these parasites. and then they again reinfect and the fever goes down. So in malaria you see this spike. The spike is basically this sudden burst coming out this RBCs and reinfect. So I was interested, we were interested that how this multiplication process takes place. Because if you can understand this huge amount of DNA replication, what are the key regulators, then definitely you can come up with some intervention processes. And I actually wrote a grant from Wellcome Trust, and without knowing anything about malaria that time. And the comments were so harsh. So
you thought that you
I thought that why even they're calling me? But they called me in Cape Town in South Africa. So I thought maybe I should go and at least I should see Cape Town, which maybe I won't go. I went there and then I tried to convince them that, see, I have experience in mammalian replication. Earlier I worked on a pathogen called Entamoeba histolytica, which causes amoebic dysentery. Now I'm going back to India and I want to use these expertise in a pathogen which is prevalent and important for India.
And it's been very, very important for India, because the kind of things that we see happening around ourselves are very disturbing. We'll keep talking about that. I have to take a break. Don't go anywhere. We'll come back soon. Welcome back to Eureka. Professor Suman, you came out from a small town in West Bengal. You graduated, then you did your PhD, and then you went abroad. And then you come back to India and ultimately land up in a university which is one of the finest universities in the country, but not very well known for sciences. You could have gone to other institutes which are very well known for doing very good work in science. Why did you choose Jawaharlal Nehru University, or for that matter, why did you come back?
So I always wanted to come back, even though I knew that
was a firm decision when you went?
Yes, because even though I went abroad and I wanted to work on DNA replication, cell cycle regulation in mammalian system, but back of the mind I always wanted to come back. And it was a dream come true. I did my PhD from there. I was a student and now I got a job there. So there was no question that I should come back and take up this job. And why university? If we don't teach in university, I like teaching very much. Where are the students going to come from? And research, no matter how much research you do in the institutes, the students have to be groomed. The students have to be inculcated for their research interest in the universities. So if we don't teach well, if we don't ask them to ask
you
this
think
question,
essentially a good scientist has to be a teacher?
I believe so.
And but we have in India a system where people like me don't get in touch with the students. Those who are working in CSIR, DRDO, ISRO, atomic energy, they don't come in touch with the students. So is there a problem with that? That is what you are suggesting?
No, I don't think I'm sure even in the institutes, you are teaching. You have graduate students, you have PhD students, you have postdoctoral fellows. They are there, you are teaching them. But maybe you are not linked with the classroom teaching directly. And as you know that why government also has started all these ICERs, 5 ICERs, because I think we realize that there is a problem in the basic education system. And unless we train these people with a good teaching facility and ask them how to ask these questions, I think we will have a problem when you go to the later stage. So we all are teaching, institute also, university also, but university is more directly linked with classroom teaching. And that many people like us, we really
That is
like
long
that.
duration acculturation
Right.
in a specific subject and specific discipline,
Discipline.
which is extremely important for doing good science in the country. You've got large number of awards, you've got scholarships, you've got grants for doing work. How satisfying that has been for you?
Well, I
Were
take
you excited when you got Bhatnagar Award?
Yes, I was, and I'm still, I am, but I think I have gone only very little. I have to go long way, and awards definitely makes you happy because this is a kind of recognition you get. But eventually, whatever you are doing in the lab has to come in the market. And unless when I'm retiring, I do see that I have contributed something which has been translated.
Which is a bigger cake for you, the award, or when you find something like you did find something novel and suddenly you realize I've done something?
I think definitely the next one, the second part.
The second one, when you discover something,
Discover
new,
something,
that is
and
the
then
big.
you want to see that what is, because many times we do discoveries, make discoveries, but follow up is much more important. Because
Absolutely.
unless you follow it up and
The rest of it is just hard work.
Right,
and
right.
Hard work. Would you like to give a message to the younger generation? You are yourself very young.
I'm 46 now. I would say that there is plenty of opportunities now. The funding situation is really good. The opportunities are good. But I think we have to work hard. If you work hard, if you ask right kind of question, and if you are focused, I think definitely the goal, maybe it will look like far, far away, but each day you will achieve something and you will be nearer to your goal.
I'm sure you would be happy to answer questions. If our viewers have any questions, please direct those questions, feedback, or any comment on email eurekarstv@gmail.com. I'm extremely thankful that you came and took time to come here
Thank you
from
very
your
much.
research. It's been wonderful. We'll come back next week with another fascinating and outstanding personality. That's all for now. Thank you.